this is a presentation. We did it last year. There was only maybe 2 people. I noticed there are a lot more here at the beginning, at least, for our session this time on hierarchical event descriptors. There's been a gap in metadata annotation of neuroimaging data, a serious one, and that is there has been no system for annotating the nature of events that occurred during the experiments. And particularly, well, for any neuroimaging, for any experiment in which alive, conscious animals or humans are participating, of course, the interest is event-related brain dynamics. And so there'll be 4 talks. I'll talk about the overview of HED and of events themselves, and event-related analysis. And then Young Trong will talk about how to actually use HED and all the tools that we've been building to make it feasible. Dora Hermes from Mayo will talk about the first HED library schema, that is add-on dictionary of terms, in this case, used by neurophysiologists to annotate EEG data events. And EEG events. And finally, Monique Dennison will apply HED to fMRI annotation. And then hopefully we'll have time for a discussion. So events and experience. So why and how is it that we recount our experience largely as a complex set of events? Sort of a hand-waving answer might be because our brain is primarily a novelty detector, as well as a memory box. It's tuned to react to recall events that were unpredicted or unexpected. As the phrase goes, turns of events in which something changed. So, for instance, as a boy, I remember having gone to the movie theater in my town and watched a re-release of the revolutionary Disney animated movie Fantasia, which I recommend finding somewhere online and watching if you haven't. It was really revolutionary and fantastic. And I remember very well some of it, although of course my memory has been changed from seeing it occasionally through my life. But going to the theater, seeing that movie and the impression it made on me remains in my memory a single isolated life event. But events, of course, are used to describe our ever unfolding experience. It's interesting, though, however, that our memories do not necessarily concern all the elements of the experience. In fact, definitely do not. So, for instance, if we saw a film, we don't remember the shots, the scenes. We remember some details, and we remember the story. We remember the effect on us. And we remember it as a movie viewing event as a whole. So, for instance, the movie Fantasia had several movements, musical symphonic music pieces on which were built animated stories, or in one case, just abstract moving patterns. So we can think then of each, we can conceptualize and remember each of those movements or sections of the film as a separate event. The experience of them is a separate event. But then each movement in this Fantasia consisted of a series of scenes in which events unfolded in the stories. And each scene was composed of several shots in which the camera angle changed. We could think of those as sub-events of the scene or as phases of the scene, and each had distinct beginning and end transition points, which we could call event phase markers, or marking transitions from one phase of the event process to the next. So within each event, and you can use the example of the movie, the section of the movie, the scene, the shot, there may be other disjunctive points in which the action or the music turns in some new direction. So each event then can be said to have at least two event phases, its onset and its offset. But it may have within it other memorable or affecting event phases. Through which, in the case of a movie, the story unfolds. So for film viewers, the onset of a new shot or scene or movement produces a more or less strong disjunctive challenge to our brain-maintained expectation of continuity in our experience. Therefore, it instigates a more or less strong brain response. That is, brain dynamic response, supporting our perception of disjunction, of novelty. However, it's quite remarkable that in our that we pay so little attention to many disjunctives moments. The prime example being saccades themselves. If one replayed the actual visual experience of a person, it would be almost nauseating to watch, because there would be jumping a few times a second. Different parts of the scene, the saccadic transitions that would be completely jarring, which we're hardly aware of. The same thing is true for a movie. I once sat on a plane on an aisle seat, and there was little TVs in the back of all the seats. So I looked up the aisle and I saw people watching different channels, and I couldn't hear the sound. So what I saw was just changes every 3 to 5 seconds to a completely different camera angle, different focused on a different person or a different part of the scene. And these were all going out of time with each other. And I thought to myself, how can they make sense of any of that, make it into a story? And yet, when you actually see a movie, you're hardly aware. You're aware only of the story if the editing is done well. And you're not aware of shots. I remember a film professor saying that he had 2 young women come to him after his freshman film class and said it was so interesting to learn about shots, that the movie is made of shots. They'd never seen that before. So I'm sort of trying to bring up the complexity of our experience, our memory of that experience, our perception of the role that perception plays in our experience, the role that disjunctions in our experience play, and of course, underlying all that is the brain dynamics that are dealing with what is important to us. So we're working on a movie, an animated movie, The Present, a short movie, very nice, that was shown to several thousand children and young adults by the Child Mind Institute in New York. While they were collecting EEG, they also showed it again while they were collecting fMRI data. And so we're trying to annotate this movie, which has been used in other projects, and all the events in the movie. So here's another example of the complexity of describing what actually happens during a neuroimaging experiment, in which one might see a movie or an animation, or listen to a story being told, or even read a story with eye movements. So for instance, this is a first-person detective story as the detective approaches the scene of the crime at night. And so the writer, he says, I went to the back of the house, I looked around, some light came, meaning he perceived that light came. I noticed. So he's telling the story here through this very sparse recounting of the various experiences he had. I opened it. I turned on the kitchen lights. Now, of course, how he did that, with what body movements and so on, what eye movements to find the light and so on, he's leaving out, but we can follow all this. It's very interesting. There were marks around the lock, which means he looked at the lock and he saw there were marks. It showed, that means he inferred. It occurred to me, he had a thought. He had an imagination. Again, he turned off the light. He waited. The house was silent. That means he was listening. And what he heard was the traffic. Well, this is very interesting to me, because it shows that with this very sparse skeleton of experiences recounted in brief through language, we can, in fact, as a reader, vicariously participate in a whole sequence of events here with associated emotions, just as the viewer of a movie can do. It's really quite extraordinary. So how can we record for use in analysis or future mega-analysis using AI, for instance, or other methods? How can we record what happened during a neuroimaging experiment? So, of course, it was seen early on in EEG neuroimaging experiments that unexpected sensory events produce notable perturbations in EEG or later MEG signals. And so the study of event-related potentials or fields became to dominate the whole field of EEG neuroimaging for decades. And they found that they were most distinct when you presented rapid onset stimulation. The word was tachistoscopic. Why? Because in 1860, someone had invented the tachistoscope, which to present suddenly pictures, probably with a spring and a shutter and so on. And then originally a falling weight to measure the reaction time. The weight would stop when you pushed a button. So this was the beginning of psychophysiology, which is now cognitive neuroscience. But offsets of sensory presentations typically have less distinctive signature in brain activity. So when became possible to record EEG and to record events that were deliberately introduced into the EEG recording session. What was recorded about those events was derived directly from the equipment that was used. So here was the equipment that I used in my PhD thesis with Robert Galambos about in the early 1980s. He had some sort of computer that could run a program. Out of the parallel port came some bit code called a trigger, and it would trigger the analog grass auditory stimulus generator. It had dials and knobs, and you dial in the frequency, and you dial in the duration, and you dial in the ramp up time and the ramp down time. And the triggers would actually trigger the analog stimulus generation. And then it would go to the ear, the brain, the EEG system, which would also get the same trigger out of the parallel port of the PC. And that was all that was recorded of the event. There was a trigger at a certain time. That's what that was recorded with the EEG system. The stimulus generator could only generate one kind of stimulus. Well, that word trigger is still used by many labs today and this concept that you record a parallel port code at a particular instant in the recording, and that's your recording of events. That's still dominating both the EEG manufacturers, fMRI also, MEG. It's dominating the experiment control programs. It's dominating the BIDS movement. It's dominating current thinking about neuroinformatics. So, so even in BIDS, which probably all of you know about by now, there are only two columns that are needed, and one is the onset latency, or the trigger latency, and then the event code, aka in BIDS values or event types. Even the duration of the stimulus event is an optional column. So what one finds when one tries to look at shared data that's been donated or shared with open repositories, such as such as Open Neuro, that's the one we work with. You'll find that this is all that's recorded. Event type 137 occurred at this such and such a millisecond. What does that mean? How can one actually analyze the data that's been carefully collected and stored in a repository? Or else some other systems might use some sort of word like target. The target was presented. What is the target? What kind of target? The presumption was always, well, if you want to look at the data, you read the paper, you look at the method section, you figure it out, and so on. But this style of analyzing, of analysis of data, of course, is being overwhelmed by in particular, the success of the AI movement, in which the best results come from large amounts of annotated data with lots of variability. So about a dozen years ago, the HED system was first conceived of, and the need for it was recognized in my lab by Nima Bigler-Shamlo. And there's three basic concepts that are now in HED. There's the basic concept of the experiment timeline, of course, the clock against which you compare all events, as well as the data collected. There are event processes which unfold through time, and there are event markers or event phase markers. So there's the onset phase, the offset phase, and in this reaching movement event, recorded by motion capture or video or something, we've put in here a course correction moment in which the trajectory of the movement changes because of new information received, perhaps. So these three basic concepts, I think, are the minimum that you have to really have a full conception of what recording an event in a neuroimaging experiment means. Events are event processes that unfold through time, and they are demarcated on the experiment timeline by phase markers, marking their onset, their offset, and then any points of disjunction within those event processes that are of interest for the analysis. And of course, the HED is a system for attaching to these time markers strings that give in as much detail as possible or feasible the nature of the marker, the event it's marking, and in a form that's both human readable and machine readable, so that it can be used in automated analysis. So, for instance, in this experiment, there was a timeline, and there were both. We see here both phase markers and event processes themselves. So here's a display of an image for a certain portion of time. Here's a reaching movement that was recorded. Here is, and this was in a task condition that was ongoing during this period of the experiment, in which the subject was to, for instance, see an object displayed in image 1, and then reach to touch it on the screen when image 2 was being displayed. For example, this is a thought experiment. So there are onset markers that are recorded, such as this. There are offset markers, like this. There are course correction inset markers, inset into an ongoing event, and then there are condition, event, onsets, and offset markers. The aim is to give a complete description of the temporal history of the experiment. Now, HED syntax is most well developed, and tools are most well developed for BIDS data sets, although we're working with NWB as well. So here is the events.tsv file. Here's the set of these triggers, their latency in the data. Here's a duration column that's been filled in. And here are some codes for the value column, are the codes for the type of event that occurred. This was from a data set from our experiments 20 years ago. And so these meaningless codes are all that was recorded. Now, we notice in the TSV file there, in the events file, there's a column that says HED. That's an optional column you can put in. And we could put in a description in HED of each of those events. But then you'd have to repeat it every time that event occurred for the same subject or different subjects. So there's a much more efficient way, and that is using the associated events.json sidecar file in BIDS. And JSON file looks like this. And what we have here is for each column type. So here's the value column. There is first of all a description of, first of all, what the column encodes for, and second, the levels are the values that occur in the column. And so these give plain text descriptions, and this is what VIDS expects. But it's also accepted by VIDS to use to make head definitions of each of these labels that occur in the value column. So the non-WM stimulus meant, in our case, a visual presentation of a sensory event across that on which the subject should fixate for a certain duration. And here is the description of the duration column, again with a text description. And since this takes numeric values, not a finite set of symbols, here the units are given as seconds, and the head string is duration in number of seconds. So either both numeric values and ASCII code values can be memorialized in the JSON file. And then at time of analysis, the head tools can assemble the head annotations by combining the TSV files with the JSON files. They can assemble complete head annotations for each of the events in the recorded, and then use that to search for particular event types or summarize the events in the data or whatever purposes. So here, for instance, is an example of a head column, a simplified example of the head column, in which we show also another innovation that we made recently that you can use in the JSON file. You can put definitions for lab jargon terms. So let's call this a recording, you say. So in the definition of recording, you can define lots of information about the recording that is now captured. You put it in the form of head tags, and now this can be captured in every analysis that uses the head information. Here is an onset of a task condition. That's again uses a definition of the reach task. We've made a definition called show image, in which the nature of the presentation as visual and so on and so forth is captured. Here is the onset and offset, here separately recorded, of the reach movement. And here is the next event on the timeline that occurs is the second image comes on, and then after that the subject makes a course correction to their reach movement in progress. And then a certain point the reach task condition goes off, and there's a define there that says more about that. For instance, it may be that some icon disappears from the screen, or there's some notice given to the subject that there's a new condition, perhaps a rest condition is occurring. Okay, so you see that a proper recounting of what happened during an experiment, even a fairly simple experiment here, where there are just static images displayed and one motion capture of some sort device to capture not only brain activity, but in this case, hand movement. So HED was proposed, and the need for it was recognized by NEMA HED is still the only system ever proposed to actually record the nature of events during neuroimaging experiments in a common language, in a way that is both human readable and machine actionable. And when we talked about it at NIH and NSF, they looked at us, blank. Even the neuroimaging, even the neuroinformatics people at that point were looked at us blankly, especially the fMRI people. Why should we want to do that? So the goal is to enable describing experimental events to an arbitrary level and then using that information most efficiently in analysis. So we had to define a syntax and then build tools to simplify its annotation. And NEMA was very fortunate to find a willing partner in Kay Robbins, the University of Texas, San Antonio, who's been really key in the further development of HED and its tool base, which continues now under the auspices of the HED working group and her small team of programmers. During the pandemic, for the last 4 or 5 years, we've been meeting twice a week on Zoom. Others can join. HED is an open source community project maintained on GitHub with tutorials and examples and so on. We finally did get NIH development support last year. The program manager said, "Oh, this is a very timely proposal." And I thought, well, what about 12 years ago when I went? It was just as timely then, but BIDS gave it the full sanction to use HED for event annotation in all the modalities, all the BIDS standards. Finally, we decided that we didn't want to have a standard vocabulary that expanded and expanded forever because it's so difficult to search and learn and use. We'd rather have a rather compact vocabulary or schema of terms with common definitions in HED, and then accept vocabulary extensions in the form of library schema that attach to the original schema or the standard schema. Now, of course, we're beginning the large job of outreach to, as here today, to explain how HED works and why one should want to use it, not only when you share data, but when you store data for your use in your own laboratory, I would argue. So here's another experiment. This is one conducted by Henson and Wakeman, and it's been shared for some years now online. So there were crosses, there were pictures of people that were shown, and then there was a judgment to be made. It was kind of a face symmetry judgment, although the actual experimental variable of interest was not the judgment of the face, but memory for the face and post-testing, and also responses to repeated faces versus novel faces, and famous faces versus non-famous faces. So although what was shared again was just the triggers from the MEG machine and not for all the events, by the way, we had to actually have correspondence with the authors and have them go dig up their original, the extra data and give it to us so that we could add it to a new version of the data on OpenNeuro. But when one actually tries to annotate what went on in those experiments, of course, there are several event processes. In fact, several implicit or explicit gaze, tasks that the subject was performed. They were asked to fixate the cross during its appearance, They were asked to inhibit blinking. They were perceiving sensory events. They were making judgments. They were making button presses and so on. And so this is example of a much more complete BIDS events.tsv file in which we've not only given definitions, used definitions for different types of events, which we define in the JSON file to show the event type, but we also provide a pointer to the actual stimulus files, and we indicate the role of those stimulus files in the actual statistical design of the experiment. So how to get to something like this, where data that is stored and shared are stored and shared in an analysis ready form like this, where one doesn't have to contact the authors, where one doesn't have to read the method sections of their various papers and then contact the authors, where one can integrate across different data sets and run mega-analysis. And this is not so easy to do, but just giving it to the 1960s level engineering to record the trigger moment and the bit code is definitely not enough for the new computational neuroscience approach to neuroimaging, which is rapidly becoming dominant for good reason. So to make data analysis ready, to share data that's analysis ready, to store data that's analysis ready, you have to report all the relevant events, including the tasks, task conditions and their timing, and then annotate them in a form that is not only human readable, hopefully, but machine actionable. So Young will show the HED schema. That is to say, the standard HED dictionary of terms, which are arranged like a forest, like in a heterarchy, a set of small term hierarchies. And with third generation HED, which we introduced a couple of years ago, this has become much more easy to use. And also with the tools that are being developed and have been developed. So HED uses a controlled vocabulary, and a formal specification is also available to guide tool building. So HED descriptions are called HED strings, and they're comma-separated term hierarchies. For instance, event/sensory event would be a term hierarchy. And then there might be an item or an attribute. Attribute would get down to eventually visual. However, because of the rule that's been introduced into HED that you can use one term only in one place in a schema, that means you don't have to, as an annotator, you don't have to annotate the entire hierarchy associated with these tags. So here's an example: event, sensory event, property, sensory attribute, sensory presentations, auditory presentation. That's the formal way of saying that there was an auditory presentation. And this event is a sensory event of auditory stimulus. One doesn't have to annotate that at all. The tools can expand that simply from saying it was a sensory event, an auditory presentation. Because of course, none of this will actually happen unless the tools make it easy enough for people to use it, and unless people realize the importance of recording the nature of what happened during their imaging experiment. What happened that perturbed the ongoing brain activity of the participants that showed them for study. So, as I said, the schema node names must be unique, and that rule makes possible the simplification of the task of annotating. So you can say that there was a circle, and you don't have to say there was you don't have to use this hierarchy. Now, why is the hierarchy there? The hierarchy is there so you can say, well, give me all experiments on open neuro in which there was MEG and 2D shapes were presented, or ellipses were presented, or geometric objects were presented, so that this helps in these hierarchies help the generalization of event searches. And then we introduced in generation 3 HED definition. So you can use your lab jargon terms. You can call something a target, but the definition can explain exactly what it was. It was a visual presentation of a cross instead of the star, or something, whatever it was. So you can use in the annotation your lab jargon terms, and then you can define them in fixed place, and perhaps for the experiment only in one fixed place, and find them in terms of head tag exactly what this target in or other term you want to use in your for your own analysis purposes, what it actually means. So if you define this, for instance, you define in the JSON file, circle only in the Hansen-Wakeman experiment as a visual foreground view of a white circle in the center of the screen with the background black. Then in annotating, you can just say, at this moment, there was the onset of a circle only event. And the tools will compose the full head string for each of those events, or the offset and so on. Okay, so library schemas. So Dora and Monique will talk about those. They extend the head vocabulary to terms that are used in a particular research subfield or field. So that keeps the head dictionary or schema from resembling the Oxford English Dictionary over time. So there's a score library that using a language of terms cited on by neurophysiologists to describe EEG data features that Dora will talk about. And Monique has been working on and has a first version of a head language schema. So for instance, what language, is it a Dravidian language? Is it a noun? Is it a verb? Is it a gerund? And so on. Terms that are really relevant for language neuroimaging studies. And yeah, so we plan to have a video, a stimulus zoo, a head zoo, where we'll have complex image sets or stimulus sets or movies that have been used in more than one experiment and have received detailed annotations. So they'll be available along with the detailed annotations for use in future neuroimaging experiments. So is there a need for a music schema with quarter notes and semitones and so on? Well, that's for the music neuroimaging community to decide. Okay, so This is a remark now that there are two opposing directions now ongoing in cognitive neuroscience, I noted. One of them is the replicability movement. Because of complaints about widespread misuse of statistics and misinterpretation of statistical results in fMRI and other modalities, there's a whole group of investigators that are wanting to make data as replicable as possible and to ensure unassailable statistical interpretations, typically with standard statistics. And so they want to declare typically minimal experiment design and the hypothesis they're going to test and the test they're going to apply in advance. So they can't be accused of fishing for a result or not paying attention to multiple degrees of freedom and so on. These investigators want to minimize variability and degrees of freedom in the data. They want to make data completely reproducible and completely unassailable. The conclusions drawn from it, the hypothesis tests, the results of the hypothesis testing, planned hypothesis testing, unassailable. That means generalizable in some way. And then there's the surging movement to make use of a different approach to modeling, and that is modeling of natural cognition. How does brain activity support our natural cognition, our experience, and our behavior? And the use of AI applied to very large, diverse, and data sets. So this group would like to model brain dynamics supporting continuous, complex experience and behavior, and train models on large amounts of well-annotated data, and in which the data richness and variation is maximized. But that richness of the experience of those experiences that the participants were having during the recordings are captured and annotated. So this was the mobile brain body imaging concept that we published about almost 15 years ago now. That we should record not just what the brain does, but since EEG in particular is portable, I suppose FNIRS and OPM and so on, their portable modalities are being developed. One could record body movements, eye movements, and EEG during complex designs that involve movements in normal 3D space, actions, motivated actions, and even social interactions. So I thought that was so important for the future that we should have a 4-letter acronym also. So I proposed MOBI. So the concept here is to simultaneously record during naturally motivated behavior what the brain does through high density recording, hopefully, EEG, or there's already been experiments of people with chronic brain implants walking around the campus, followed by a video cart and an EEG cart and so on. What the brain experiences through sensory scene recordings and, of course, recordings of the experiment control program, and what the brain organizes, and that's our behavior. And then use math, basically, to find, model, and measure non-stationary context and intention dependent functional relationships between brain activity and all these experiences, all this experience. And I believe still that this is the future of, this direction at least is the future of neuroimaging, of cognitive neuroscience. And there's now a wave of tens of thousands of data scientists who are being trained, and some of them are going to be interested in how the brain works. So we're now at a point where we have HED has a very good and compact system for representing simple events, beeps and boops and pictures shown for a finite amount of time. Too short for the subject to have eye movements unless you record the eye movements. So the experiments of the past, HED can now, the experiment designs of the past, I'm sorry, those are experiments, of course, still ongoing. HED can capture those very well, and the tools to do so easily and to exploit that information are now available. But for annotating natural cognition, what did you experience in the movie? What did you experience in that conversation with another wired up subject? All that kind of natural cognition paradigms. There are lots of challenges in recording the nature of experience. In particular, even in standard psychophysiology experiments, tools to use information about preceding events and about priming events. Software tools to use that and enter that in the analysis are still missing, largely. HED doesn't yet have a syntax for describing relationships between events. For instance, whenever you see the red A, push the button. Well, that's an action imperative. The A is imbued by the task with an action imperative to press the button. So we don't have the syntax or the tools to capture, to record, to annotate the nature of participant intentions, including task designs. No one has yet made a computer language in which to express task designs, experiment designs in a common language. It's a very important task, I think, for the future. HED doesn't yet have much syntax to record spatial relationships, because when we think about doing that, we record, well, there's relationships of things happening on the 2D screen. There's a relation of the participant to the screen in 3D. How far away are they? Are they moving their head? Are they moving their eyes? There's the relationship of the 2D screen to, for instance, a movie that shows an imagined or a represented 3D space and objects and agents within that space, let alone when someone has a virtual reality headset on and is wandering around in a virtual space or in actual space, performing a MOBI experiment. So there's lots of challenge in annotation of events in natural cognition, experiments that are capturing more of natural cognition. Capturing body position, body movements, as well as terms used in other subspecialty research specialties, social, aesthetic, affective cognition, music cognition, and so on. One can actually look to the future to actually see world knowledge being included in the analysis. So if the subject is walking through town, with implanted electrodes, the analysis could have all the existing knowledge about GPS and otherwise of that town, what it is they're experiencing. That is not very far away, actually. Impossibility. And of course, we'd like to connect HED to one or more brain atlases, so that the analysis can proceed in a larger so HED can facilitate use of a larger range of activity, and the tools that use HED to in analysis of brain data can have available immediately various types of knowledge. So that's an overview. The nature of events is much richer than the 1960s triggers coming out of the parallel port. But the field somehow has fixated their thinking on that antiquated model, I should say. So using a better model of event processes, event phase markers, and developing a syntax for that, and very importantly, developing tools to make it easy to express the nature of experience in the experiment in those terms. And then tools to use that to search within data sets and across data sets for events of various sorts, or to feed entire sets of data sets into mega-analysis programs for various purposes, including sort of AI-driven. Now, I'm putting AI as a shining beacon here. We know that AI models are now very poorly, if at all, interpretable, and that is an obstacle to scientific understanding and progress in scientific understanding that are being addressed now more and more intensively by mathematicians. However, here we stand at this junction point where there's more and more conscience and mandates to share data, to make it available for these new types of analysis and reanalysis, further analysis, and mega-analysis. And to do that, one needs the data to be analysis ready. And crucially for brain data, one needs of awake, aware organisms, including man. One needs to record what happened during these, what did the participant experience, moment by moment during the experiment, to the extent possible to say that. Without that, progress in neuroimaging will be continue to be slow. So now I'm happy to answer questions, and then we'll have Young speak on how to actually use HED. So is there someone to moderate?
I can help with that. So if anyone posts questions in the chat, Scott, I'll read them out to you.
Well, one is, of course, one can go to the HED GitHub site and post issues and questions there. You can send us email. You can join the working groups.
Yeah, Scott, can you drop the link to that in the chat on Gather?
Yes, I'll do my best to do that.
Awesome, thank you. If anyone has questions, you can either write them in the chat at the bottom right-hand side of your screen, or you can come up to either side of the stage to the little footprints and the microphones.
The website is headtags.org. Well, I think in the interest of time, since I went on quite a ways, let's move on to the next presentation, which should be by Young Trung in a more practical vein, how to actually use the tool infrastructure surrounding HED to annotate your data and to make use of those annotations for analysis.